All right, we're talking biochemistry and we're talking about irreversible inhibition. We got three types of irreversible inhibition: group specific, affinity labels, also known as reactive substrate analogs, and suicide inhibitors, also known as mechanism-based inhibitors. So how are you going to remember the three types of irreversible inhibition? You're going to remember that Andrew has irreversible gas. Gas being an acronym for the three types of irreversible inhibition. Is it gross? Sure. But if it helps you remember, then it's totally worth it. Real quick, let's cover the key characteristics that we see going on with these irreversible inhibitors. First of all, and probably the most important thing to understand, is that these inhibitions that occur are permanent. And that's why it's called irreversible inhibition. So these inhibitors permanently inactivate the enzyme. And this permanent inactivation occurs through strong chemical binding. So these inhibitors make strong chemical bonds with the enzyme at the active site, and these are usually covalent bonds, which we'll talk about. Now, because the enzyme is permanently altered to be inactive, increasing the substrate concentration is not going to overcome this inhibition. So you can't just add more substrate or remove the inhibitor to reverse this inhibition. It's not going to happen. You need fresh new enzyme that's not been altered and modified to be permanently inactivated. Okay, let's start with the G in gas, group-specific inhibitors. So these inhibitors react with specific functional groups that are in the active site of the enzyme. And through this reaction, the inhibitor forms a covalent bond in the active site of the enzyme. So take a look at this example on the right of the screen. You've got a hydroxyl group, the OH. That's your functional group at the active site in the enzyme. Your inhibitor, which is in blue, is going to come by and react with that hydroxyl group that's part of the serine residue in the active site of the enzyme. And so you can see on the far right, you have an inactivated enzyme because that inhibitor has now formed a covalent bond with that oxygen that's part of the active site of that enzyme. And so you've got this big chunky inhibitor thing there that's taken up a lot of room, but it's also covalently bound to the active site. So that active site is no longer active. There's nothing that can come by and bind to that active site anymore. So that is group-specific inhibition. So now we come to affinity labels, also known as reactive substrate analogs. So these inhibitors have a similar structure to natural substrates that these enzymes would bind to anyway. So these inhibitors are reactive molecules that are analogous to the natural substrate. I like to think of them as good twin, evil twin. You got the good twin, which is your natural substrate that this enzyme would bind to anyway. And then you have the evil twin. It looks like the good twin, but deep down, it's not. It's very evil. It's going to inhibit this enzyme's activity. So it's the evil twin. And the way these work is they contain a reactive functional group. And it's this reactive functional group that's going to make a covalent bond with the amino acid residue at the active site of the enzyme. So let's check out this example. First, let's establish our enzyme. Our enzyme is chymotrypsin. That's in yellow. And chymotrypsin is a digestive enzyme found in pancreatic juices. And then on the left of the image, you've got your good twin and your evil twin. Good twin is your natural substrate that's going to react with chymotrypsin as it normally would. And then you've got your evil twin. And it looks a little bit like the good twin. It can pass for the good twin, but it's going to inhibit this enzyme, so it's the evil twin. Now look at what the good twin and the evil twin have in common. We're looking at this specificity group in blue. This is what allows the good twin and the evil twin to bind to that histidine residue in the active site of chymotrypsin. So on the surface, the good twin and the evil twin, they look a lot alike. They pass for each other. And so this specificity group that allows this binding, that's the affinity part of the name affinity labels. So the evil twin looks like the good twin, and so chymotrypsin still has a high affinity for the evil twin because it thinks it's the good twin. As far as it's concerned, they're the same person. Now take a look at the reactive group in red on the evil twin. The good twin doesn't have that. This reactive group, that's the reactive functional group that's going to make a covalent bond with the amino acid residue, in this case the histidine residue, in the active site of chymotrypsin. So when this functional group gets attached to this residue, that's the label. This effectively, quote unquote, labels that particular enzyme and renders it inactive. And because we've got this strong covalent bond, it's permanently inactive. And finally, we got suicide inhibitors, also known as mechanism-based inhibitors. Now, suicide inhibitors work a little bit differently. First of all, they start out as inactive, but they can still bind to the active site of the enzyme, and once they do, that binding is what actually activates the suicide inhibitor. So now that this inhibitor is activated, it makes a covalent bond with the enzyme at the active site. That permanently inactivates the enzyme. Here's a visual representation. And because I didn't plan ahead, I don't have a microphone stand. So my microphone is going to be the enzyme. This stress ball that I hope you remember to take to all of your exams, this is going to be your suicide inhibitor. So the suicide inhibitor binds to the enzyme at the active site. The enzyme modifies and activates the suicide inhibitor. Upon activation of this suicide inhibitor, a covalent bond forms between the suicide inhibitor and the enzyme at the active site. So it just stays there, and it's covalently bound, so it's not moving, and it blocks anything else from binding to the active site. So that's why it's called suicide inhibition. By interacting with the inhibitor in the first place, the enzyme is effectively sealing its own fate by hitting the self-destruct button. So that's irreversible inhibition. If you have any questions, let me know.